First, thank you for the invitation to be here. It's a pleasure to be here at this committee. So my talk is going to focus on biomarkers and some emerging biomarker approaches and technologies for looking at associations between environmental exposures and adverse health. Just as a quick overview of what I'll be going over this afternoon, first I'll talk a little bit about environmental exposures and biomarkers and how the two of these fall within the continuum of exposure and disease. I'll talk about the concept of the human exposome as a complement to the genome, and then I'll talk about targeted versus untargeted biomarker approaches and what are some of the advantages and disadvantages of these approaches and how they can be used to work together to look at associations between exposures and health. Finally, I'll finish off with some field-friendly biospecimen collection devices. So once we have these biomarkers we want to look at, how can we get the blood samples to do these analyses? And there's some emerging technologies there. I did put up a disclaimer that one of the examples I'm going to be giving is a heavy metals-free blood collection device that we're using right now with InMed Microanalytics for looking at heavy metal exposures in newborns that I have a financial interest in. So to begin with, I just show this continuum of exposure to the disease. And so we have environmental source, transport, exposure when you come in contact with a human, internal dose, the amount that gets into a person, biological effective dose. This could be a metabolite of a compound that needs to be metabolized to exert its toxic effect. Early biological effect, altered structure, function, and disease. And along this continuum, there's a lot of different places where exposure can be assessed. And it really comes down to what are the questions you want to ask, what are the relationships that you want to look at. We just heard about a lot of these different emerging techniques for environmental measurements, where if you're going out to the source and where these are being transported, you can use things like satellite imaging, personal monitors, wearable devices that can assess exposure outside of the body. But the main thrust of my talk today will be talking about biomarkers, and in particular blood biomarkers, once these get into the body, where we can measure them. And I think one important question to think about as well is along the continuum, what time frames are most important? Because we talked about when a biomarker first gets into the body, it has a biological half-life, so you got to collect it at the right time. There are also response biomarkers that respond to the exposures that can help you look at underlying mechanisms that are going to help link the exposure to the adverse health outcome. And so there's a lot of questions along the continuum in designing the study of what types of markers would be best. So just real quickly on, and this got mixed up a little bit, on the human exposome. This is off of a paper of Steve Rappaport and Martin Smith that came out in 2010 talking about this concept of the human exposome, where in this paper there was actually 2 National Academy of Science meetings that focused specifically on this concept of the exposome as a complement to the genome. I think most important here is some of the take-home messages with this is that we need to take a more broad approach when we look at exposures. That it's not just some of the chemicals that we can measure in air and in water and in food, but we have to look at our entire internal chemical environment, some of the things that we're exposed to, the reactive chemicals that are endogenous that might have causal effects with adverse health outcomes. So now I'm going to switch gears. I'm going to talk about both targeted and untargeted biomarker approaches and some of the advantages and disadvantages of these. I'm going to start with untargeted biomarker approaches, and these are your omics approaches, so global approaches for going in and being able to find things that you don't need to know you're looking for before the experiments happen. And we had some really nice examples of this with metabolomics that we saw yesterday. I think this schematic just kind of shows some of the key features of untargeted analyses when you do this. Often you do this in a case control manner, and so you would take 2 different sample types, exposed, non-exposed, or diseased, non-diseased, and you're looking for unique features that might tell you something that is unique about the sample and might be driving the health outcome. High-resolution mass spectrometry is commonly used because these types of experiments often have thousands of features that you get out of them, so you need to have the high-resolution mass spectrometry to tell differences between masses. I will talk about one other type of omics approach that we do in my lab, which we call adductomics, which is a complement to metabolomics, where metabolomics are all the small molecules that you find in the blood, and you can do this in an untargeted fashion for looking at these small molecules. However, a lot of the molecules that we want to look at in our lab are very short-lived, and so you can't measure these directly in the blood. And so these really reactive electrophiles, what we do is we extract abundant blood proteins. In particular, this is human serum albumin, the most abundant blood protein in plasma. And there's one highly reactive site, the Cys34, that acts as a sink for binding these reactive chemicals. So we extract this protein, and we look at these addition products or adducts that are formed on the protein as a complement to metabolomics. And like metabolomics, you can see on the left-hand side in red, it captures exogenous chemicals, so pollutants from the environment, but it also captures a whole host of different compounds that are endogenous in nature that allow you to look at relationships between exposure and response that can lead to mechanism. And so first, I wanted to put up this slide to just talk about some of the challenges of using untargeted biomarker approaches. And this is a slide I borrowed from John Sobis at the EPA, who was a colleague of mine, who ran this study and TACT. And what they did here is they wanted to take untargeted biomarker approaches using metabolomics and put together blinded samples. So they took toxcast chemicals. These are really well-defined chemicals, and they made up chemical mixtures, and they did spike these into some different matrices as well. So dust samples, serum samples, and wearable wrist bands, and they sent them out to some of the better labs in the country to see how the results would come back and how they would differ. So here, I want to highlight the serum, which I'm focusing on the blood biomarkers. This was 95 different chemicals that they spiked into these compounds, gave them to 7 different laboratories, and had them do their untargeted metabolomics on them. And what you can find very interesting is some of them did a very good job. The top one in green shows 92 markers out of the 95 that they detected. Some of them had a lot of false positives. You can see some labs in pink, up to 614 features they detected, and others missed quite a few of them, where they're reporting somewhere down in 50 rather than the 95. So the take-home message with this, it wasn't that the labs were doing a terrible job at this. It's just when you're doing untargeted analyses like this, you often use different protocols, you use different instrumentations, you have different questions for the space that you're kind of using to look for these markers. And so this is often a better platform used for discovery when you're trying to look for new things, but for quantitation, it can have some challenges. And it's not new to the field of metabolomics. This is a paper that came out in Nature Methods for proteomics, and they're making the same story. That traditionally in proteomics, the idea was how deep can you go in the proteome? How many different features can you measure in an experiment? And in this paper, they started to make the push forward the idea that it might be better to do targeted analyses, that once you know what you're looking for, and you can go in and look at these at better concentrations, at lower concentrations, that is, better quantification and higher throughput, it really powers you to be able to look at these in population-based studies better. And so this is another schematic just showing some of the differences between a targeted method versus an untargeted method, untargeted platform. And some of the main key changes here are, as you can see, we have internal standards in the front. So whether it be perfluorinated compounds, PAHs, that's naphthalene. Once you know what you're looking for, you can put internal standards into your mixtures. You can have calibration curves, which allow you to have better quantification. You can choose different analytical platforms, which if you don't need the high-resolution mass spectrometry, often you can have better sensitivity and you can have higher throughput, which we're finding with some of our assays, which might be more appropriate for large-scale studies when you're ready to ramp up. And so going back to the protein adducts, this is just an example of a targeted assay with protein adducts when you know what you're looking for. In this case, it's benzene exposures. When you know the chemical of interest, you can make a very sensitive assay where here we have benzene, we know it gets metabolized to benzene oxide and the quinones. And if you look at the adducts that are formed with benzoquinone and 1,4-benzoquinone and benzene oxide, and you plot them against the air concentrations, you see that you get this nice dose response. So whereas some of these are not going to be showing up potentially in in a discovery experiment, once you know what you're looking for, you can really get better sensitivity for looking at these in targeted assays. And I just want to use this study by Stan Hazen, which I think is a really good example of how the two platforms can work together. This was a paper in Nature in 2011, and Stan's group wanted to look at risk factors for cardiovascular disease. And so they took a group of people who had events, so they had heart attack, stroke. They went back in time and got specimens, which they looked at untargeted metabolomics to try to see differences between the groups. They identified 18 different unique features between them. 3 were highly correlated, showing that they're part of the same biochemical pathway. And then they figured out that these were metabolites of dietary lipids. In the second phase of this, they went to a second study, and in a targeted fashion this time, they measured about 2,000 people. So these 3 different compounds in 3,000 people, and just quantified the 3 biomarkers of interest. Here they looked at the odds ratios for these, and what you can see here is that the odds ratio for these 3 compounds that they found were way up there with like heavy smokers for cardiovascular disease. So it was a big finding at the time. They then went further, did animal studies, and they found that there's this gut-flora interaction where these phospholipids that came in from dietary sources first need to be metabolized by gut flora, which trimethylamine oxide then got into the blood and was this big risk factor. So I think this is just a really good example. If you could start with untargeted analyses to find the biomarkers that you wouldn't know to look for, targeted approaches that you can go in and quantify these in a larger number of samples, which ultimately can lead to mechanisms for remediation. So next I want to talk a little bit about how to get the samples. So we talked a little bit about the 2 different types of approaches, untargeted and targeted biomarker approaches for quantifying the biomarkers. But first you need to get the blood specimens. And typically venous blood is better. You get a lot more of the blood. They're better defined. But these are not always possible in field settings. And so our lab uses dried blood spot sampling to obtain dried blood spot samples when venous blood is not available. Some of the advantages is that you do not need a phlebotomist to collect the blood sample. And in fact, we're involved in a couple different studies right now where we're having our participants collect their own blood sample, where you can send a kit out to them. They can prick their finger. They can put the blood onto filter paper and let it dry and send it back to you. It's less invasive. It doesn't need to be processed in the field like serum would be or plasma would be. And it's easier to transport and store, which goes down to lower cost. This is just an example of a study that I did where we got blood samples in Slovakia. These were steel workers that were exposed to polycyclic aromatic hydrocarbons. And just an example of if we could have done dried blood spots in this study, it would have been so much easier to not have to transport this box with 100 pounds of dry ice in it to bring the samples back to our lab. So some of the challenges of doing dried blood spot sampling, however. The first thing, of course, is minimum blood volume, that if you have a Vacutainer over blood, you've got 5 milliliters or more blood, where a blood spot is about 50 microliters of blood that's dried onto the filter paper. So your techniques for measuring the biomarkers have to be very highly sensitive. Some biomarkers are not stable, so if you have something that's volatile that's in the blood, once the blood dries, those go away, so you can't measure those. And you have a more complex biological matrix, that when you have a dried blood spot sample, the red blood cells will lyse when the blood dries, and now you have all the content of the red blood cells that you've got to compete with when you're doing your analyses in the sample. Some ways to get around this are our commercial products available, hemoglobin, where you can take a dried blood spot, you can elute it, add hemoglobin, which binds to the hemoglobin, and you can centrifuge it out and pretty much get back to a plasma sample. Prospectively, this is a group we're working at the University of Washington, and here, if you don't already have the samples, in this case, you can take this new paper, which has chromographic properties to it. You can apply your blood drop to the one side of it, and the chromographic properties of the paper will pull the plasma in front of the red blood cells, and then you can dry it. So essentially, you can get a dried plasma sample in the field, which for many assays is much easier to deal with. So what can we measure in dried blood spot samples? Jeff Freeman, who was a colleague of mine at applied physics labs at Hopkins, had this really nice review paper that he put out last year where they did a review of all the different chemicals that have been measured in dried blood spot samples, and there were over 2,000 analytes, about 50% of them small molecules that were reported, which I think is a really exciting paper. At the same time, I think we need to make the distinction between detecting something in a sample versus being able to quantify it at a high level, that these are not 2,000 chemicals that we can accurately quantify in samples. These are things that have just simply been detected. This is a review paper we're putting out right now that just looks at some of the exposure biomarkers in dried blood spots that have validated methods. I know it's a little hard to read, but I know if you look on the side, things like benzene, DTT, PCBs, biphenyl A, perchlorate, these are all types of chemicals that might be of interest, where there are targeted assays that have been developed for these in dried blood spot samples. Heavy metals is something that we've been doing in dried blood spots for quite a bit, so I'm going to talk a little bit more about those to show you kind of what needs to go into an assay if you do want to optimize it for a specific class of chemicals. This is work that I started almost 10 years ago with US EPA. When I came to Northwestern, we got a grant to do this through that National Children's Study, where we got blood from babies at the hospital. We got matching blood samples from finger prick samples and from venipuncture. And we looked at the concentrations of lead in the blood spot versus the venous blood draw. And you can see the correlations are really poor with the red little triangles when you're looking at the blood spot versus the whole blood. And we try to do a correction where you could take a background subtraction of the filter paper next to the blood, and that's what you see in blue, and you also really poor correlation there. What we found is you have to go in and you need to remove contamination from the filter paper if you want to get a better, more precise measurement of some of these metals that have issues with contaminations in the filter paper. So we did this. It worked really well for lead. This has worked well with arsenic, cadmium, and mercury, and we're also expanding our elemental panel right now for other metals that can be quantified in these dried blood spot samples using the pre-treated filter paper. There are also omic techniques that you can use in dried blood spots that we've been doing for a while. This figure actually comes out of Dean Jones's lab at Emory as well, and it just shows kind of the relationships with some of these omes that we heard about yesterday. Starting from the bottom, you have the exposome, the metabolome, the cysteine, proteome, and this would be where my adductomics kind of fits into, and then how it relates to other things such as this transcriptome and epigenetics. I think more and more, as we have studies that pull in these different types of techniques, we'll have to use the types of analyses that we heard about yesterday to start bringing in the data to kind of figure out what's going on the systematic level. Yesterday we heard about how the data can be used together in some of these studies, so I just wanted to end by showing a couple slides of what we're doing in the laboratory and with our devices to try to be able to collect these to get multiple omic measurements in single samples, working with universities, a couple different universities. We've come up with this new technique for field-friendly blood collection where we can take a dried blood spot sample, take a single punch of a dried blood spot, which is about 1/7 of a dried blood spot, elute it to do adductomics work, and we can get enough protein out of that one little 7 or 3 millimeter punch of the dried blood spots to do our adductomics. But then we can also spin the small molecules down through a molecular filter, and we can perform the metabolomics in the same exact sample, so we don't have to use 2 samples for this. And these were done at Mount Sinai with our colleague Lauren Petrick, who did this work for us. And so this is allowing us to take a single sample and really start to expand the types of things that we can measure. And I'll also note that we could apply both targeted and untargeted platforms in both of these analyses. To finish up, I just wanted to talk a little bit more about the devices. I talked about this at the very beginning a little bit more, that we're using this for newborn screening. But as we have specialized biomarkers that you know you want to measure, you can improve upon the devices to get the blood samples that you need to perform these analyses. So here we have our pre-treated filter paper that's built into our device that has the metal-free surface that doesn't have the heavy metals. The case is designed so that it protects the filter paper before, during, and after blood collection, which we found is very critical, that it's very easy to contaminate a sample after or even before collection, after we wash the filter paper. And then there is an integrated built-in desiccant that goes into the system so that we can collect the drop of blood, close it immediately, and the blood is dried within the device. And so this is what we're using for heavy metal screening right now, that we're using with newborns, but could have field applications as well. We're also working with LifeWare Labs, and they're coming up with additional platforms for collecting blood in the field. And these are things like little Band-Aid devices with microneedles that can take small amounts of blood that you wear, and they can perform either analyses in the field or collect onto filter paper, where you could send them back to the lab and do a lot of these same analyses that we talked about this afternoon. So these are just a couple examples, specialized devices that might make it easier to get the samples needed out in the field. I know timing of when you need to get the samples is often key, and even repeat sampling. And so I think having some additional options to be able to collect blood in the field might be nice to have. And I think that's the mic.
Thank you very much. The technologies and tools are evolving. Great to hear. Questions for Dr. Funk? Sverre?
A couple of things about adductomics.
Yeah.
One sort of an information one. The difference between protein, just plain old protein adduct analysis versus protein adductomics is that you can characterize an array of adducted proteins in the latter case versus, say, just albumin adducts or something.
Right.
That's essentially the difference.
It is. So there are different proteins that you can look at protein adducts on. They give you different levels of information, like you can measure them on hemoglobin versus HSA. This platform of Adductomics, which is our more global platform, has the advantages of being you don't have to know what you're looking for ahead of time. And there's big challenges. It took a lot of time to get this assay to the point where we could go in and look for hundreds of different adducts in single samples in a true untargeted manner. We haven't been able to do it with hemoglobin yet, but the possibilities are there, and we are working with that a little bit.
The second question is, I mean, you introduced this with, we all know the sort of issue of biomarkers and, exposure biomarkers, response biomarkers, and sometimes it's a little fuzzy border.
Right.
With the adductomics, I know you had something on the list there, but give me an example of something you would characterize as a response biomarker that you would get from adductomic.
We're seeing some really interesting ones. So we're seeing direct oxidation products, so the addition of 2 and 3 oxygen molecules. We're seeing small disulfides that are binding, so antioxidant capacity. Crotonaldehyde is one of the ones that we've seen in a lot of samples, which is a marker of lipid peroxidation. But we have some new data coming in where we're looking at newborns, actually, and we have estimates of air pollution exposures in the mothers, and then we're collecting the newborn dried blood spot at birth. And we're seeing differences in several of these adducts at birth from exposures that occurred during pregnancy. So we're seeing that they're picking up on a lot of, I think, underlying pathways and perturbations that are going to be really neat to follow up on.
Could I turn Sverre's question around a little bit and ask what the limitations of this system are? What are the exposures that are too ephemeral or that we don't have the technology at the moment to pick up using these more cheap and dirty techniques?
Yeah, that's a great question. I would say one of the differences between our untargeted platform right now and our targeted platform is that we're really picking up on a lot more of the abundant things. If you look at the blood exposome, so all the different chemicals that you find in blood, there's a fairly recent paper that came out that characterized this, and they put them into 4 different categories of exogenous exposure, so pollutants, diet, drugs, and endogenous processes, and took all the chemicals that have been found in blood. And of course, there's a huge range of all of these in blood, but what was interesting in the paper is that the pollutants from the environment, the exogenous ones, were a good order of magnitude lower in concentration than the other ones. And so I think one of the challenges when you're doing these discovery experiments and looking at everything at once is a lot of times these low abundant markers aren't showing up like they could in a targeted analysis. And so it could mean we need to go to higher exposed groups when we're doing our discovery experiments, and then potentially we can take these candidate markers and drill down further with a targeted method where we could be able to measure them. But I think that's one challenge.
If you wouldn't mind, if you could provide us a reference for that paper, that would be very helpful. Oh, we've got it. Thank you.
So biomarkers are always an interesting question. You find something and then try to figure out, does it predict? And so when you look at the newborns, particularly at the lead that you were talking about, what happens if have you ever done any follow-up to look at 3 months later? Does that biomarker persist? Is it gone? What is the significance? Obviously, lead's of great concern, but
right.
What are you finding?
That is a question I'd love to know the answer to. We are actually doing this right now. That at Northwestern, we're in the planning stages to start screening all of our newborns for heavy metals, and lead being one of them. And then the idea would be to follow them and watch health effects and to see what kind of associations we see with these very early on exposures where not a lot is known, how they impact health. But that is something that we are doing right now.
So, other questions? If not, thank you. Very, very exciting work.